Okay, first of all, I would like to thank the organizers of this meeting to give me the opportunity to speak to you all. I actually think I have the easiest job of all the speakers here, because I was asked by the organizers to put all these ethical issues and considerations into a more technical background. So basically, whereas I think everybody else will have to start speculating about what will happen tomorrow, next week, and next month, I can actually talk about what happened the last 45 years or so, which in this context is definitely a lot easier than predicting the future. What I've been trying to focus my talk is on first of all, trying to get an answer, are these editors really such a big deal? Well, if the answer is no, I guess we can all pick our bags and go home. But since we're all sitting here, I do think the answer to this is going to be yes. And I want to make it clear why these editors are such a big deal from a scientific point of view. And for this, I need to discuss how these editors work, but also how we as a scientific community have been making mutant animal models in the last 40 years. And only by comparing that, I think, for the non-biologists, it will become clear why this is such a big deal. And in the end, I will spend a couple of minutes on kicking off the whole ethical discussion. I do like to point out that I'm myself a biologist, I'm a scientist, I have no ethical training whatsoever. So the angle from this part is purely my personal viewpoint, and this is coming from using genetically modified animals for 20 years. People with ethical training might think completely different about this. So before we start, for all the non-biologists in the audience, I have to make one thing clear, and that is, we biologists, how we can do things is because we steal everything. And we are not very good in coming up with things that are really completely new. Almost everything we do, we steal somewhere in nature from bacteria or viruses most of the time, and we put them in a different context, and there we can use them for either the same thing or for something completely different, but in the end, almost everything comes from elsewhere in nature. And because of that, I will have to give you some hardcore developmental biology backgrounds and other technical aspects. So for that, I hope this will all be clear. So the first age in getting animal models, I would like to call the age of breeding. And then, of course, we're all thinking of this. I'm a cat person. So if somebody with a big bag of money would come to me and he or she would say, can you start with this and turn it into this? Maybe that. I definitely don't want that. that person would give me a lot of money for it, and despite having 20 years experience making modified animals, I wouldn't have a clue where to start. Nature has been pretty good at that, and that's because of a thing called selective breeding. Of course, these are in the end all coming back to the same species, and all these different the dogs we now know have only been getting there through selective breeding. It's also being used in other things. For instance, here we have some of the improvements that were made to agricultural animals over the last couple of decades. And I should point out, this is not completely because of selective breeding, but this has been a big part of it. And as you can see, all the different things can be improved or changed quite significantly without any genetic modification at all. And even closer to the biomedical sciences, the field where I come from, is we have, and of course nobody will be able to read this, and that's exactly the point of this figure. We are using all these fantastic mouse inbred lines, and of course also the case for other species. All these mouse inbred lines They have different behaviors, different phenotypes. Some of them are very prone to get cancer, others for other types of diseases. There are a lot of spontaneous mutants that are extremely useful still in research. But in the end, this is almost one big pedigree that all goes back to the same place about a century ago, coming from people who were just breeding mice for coat colors. And by simply picking particular animals and continuing breeding with those, again, nature, with a little bit of help, came to all this enormous varieties of mice with different phenotypes. So we don't need, by definition, a genetic modification to come to different animals with particular phenotypes. And an advantage of this selective breeding, if you think about it, it's surprisingly efficient. Just think back on all the different types of dog breeds we got in the 10 or 15,000 years, maybe even less since the dog was domesticated. On the other hand, 10 or 15,000 years, I don't think there are going to be many funders who are going to put money into that. It is relatively slow, of course, and it is completely unpredictable. You have no idea what's coming out. And you will also pick up different changes in multiple characteristics. For instance, if you would want to turn the wolf into a chihuahua, but it should still howl instead of getting yelled, you might not get it. In the end, it's completely and utterly random, of course. So the more scientific approach, which is of course why it takes us back to the life cycle. And this in particular thing is the mouse life cycle. I'd like to point out, I'm not just mainly talking about mice here because that happens to be my favorite model species. As you will see in the last few decades, for purely technical reasons, the mouse has been the species where technically by far the most has been possible. So for this reason, until now, it's mainly mice. This is sort of the life cycle for the developmental biologist, where in the end, of course, once the animal is being born, the exciting stuff has happened. And then after that, the only thing that still needs to be done is passed on the genes to the next generation. And to do this, of course, we need to follow back the germ cells. And if we see the very first progenitors of the germ cells, in the case of the mouse, are found at 5.5 days after 5.5 days of development. Now, this is important because if we want to make a modified animal, and we don't want to have to do that all over again and again every couple of weeks, but we want to get these modifications in the animal's germline, we have to do this before this point where the first germ cells appear. Everything we do before that point, so in those first 5.5 days of development, if we're lucky, it also ends up in these earliest germ cells, and then it will go through the whole cycle. If we do things afterwards, a chance of getting it into the germ line are pretty much zero. So these are the important stages of development if we want to modify animals. So if we look at in a bit more detail, and again, this is mouse material, and also the timing here in this is about mice. We start with the zygote, the fertilized oocyte, and initially things go relatively slow with cell divisions up to there we are, the 8-cell stage. At that point, something happens that's called compaction. And where we start here with a really beautiful embryo, there's still 8 clearly recognizable individual cells. Over the course of a few hours, these cells start to become some sort of amorphous mass. You can't really see the separate cells anymore. And this keeps on developing, and then at about 3.5 days, we start to get a cavity in the embryo, and we form what's called a blastocyst. And this is a hollow ball with some some cells, I'll come back to those in a couple of slides, in the middle. So this end stage and the begin stage are indeed the stages that have always been used, well, always since the 1970s, to make genetically modified mouse models. And the earliest stage, the fertilized oocyte, has always been used for the generation of normal transgenic mice. Of course, for somebody like me who's been doing this for 20 years, it's relatively straightforward. And for the non-biologists, this might already look rather frighteningly. But in the end, everything we need to make a transgenic animal that expresses something that it normally doesn't express, either something from a different species or even something from the same animal, but at a time or in a tissue that it normally doesn't do. In the end, everything that we need is a piece of DNA, which contains the part that we actually want to express, obviously, and some other parts that will determine exactly where this particular gene is active or is expressed. In the end, that's all what this comes down to. And this, again, I put this in for the non-biologists. I still think this looks great. You can hold these early embryos with a sort of pipette, and then you take a very sharp needle, which has the DNA in it, and you just prick it in. It's coming again. You find the nucleus, and you can inject the DNA this way. I see Carlisle laughing. He must have done this for a couple of decades, and he still thinks it's fun. And so in the end, this is everything that needs to be done. After that has happened, the DNA that you, the piece of DNA that you inject can integrate randomly into the host genome. This is again a chance process. And after this, the embryo is put back into a pseudo-pregnant female. And this bit of DNA, if you're lucky, it's being expressed where and when you want it. And if you're lucky, it's being passed on to the next generation. So this technique, which has been around for 3.5-4 decades by now. The good thing of this is you only need your piece of DNA of exactly the things that you want. You don't need to know anything about the genome of the species where you are injecting in. You don't need the whole sequence. You don't need the actual material. You only need what you want to put in. And for a long time, that has been a big advantage of this technique. It's also an important advantage, and then especially we will be coming back to that this technique is possible in every species where you can take these early embryos, inject them, and put them back. You don't need anything else. And it's reasonably efficient. I think about 5% of the founders, which are the animals that are initially being born, will express the transgene and will pass it through the germline. Of course, 5% when you're dealing with species that have a lot of offspring, for instance, zebrafish, if you have a clutches of 150 embryos, you will always get them. If you're doing this with sheep where you get 1 or 2, then you still have to be pretty lucky and generate a lot of animals. It's quick. You only need to inject, which will take a couple of hours for the whole experiment. And after that, the normal biological timing takes over. And we can change a lot of things these days, but we cannot speed up the embryonic development. It's mainly used to express or activate particular genes somewhere and exactly when we want it. And since it's a random process and because of a thing called homologous recombination, this piece of DNA that you put in, it doesn't integrate in just one copy, but it starts to make these long chain of dozens, sometimes more than 100 copies of this, and this whole chain will go into the injected genome. Because of this high copy number, you can get massive overexpression if you want to. The cons for this technique are that it's still a random process. It's random integration, and this exact copy number that you get is completely random, and because of that, it is unpredictable. And it's also, you can take the same piece of DNA and inject it all over and over again. For some construct, you will always get the same effect, but that's only a few of them. For most of them, it completely depends exactly where it ends up in the genome, in what copy number, and that will determine what you will actually get. In many cases, you have to be lucky to get more than 2 lines that behave exactly the same. The last disadvantage is the high copy number can give you a massive overexpression, which of course was also an advantage. And that indeed completely depends on the type of experiment that you want to do. If, let's say, you want to make an animal that makes some sort of new protein and puts it in the breast milk, you want massive overexpression, because then you get much more of your protein. If, however, you're looking at a much more subtle biological process with a too high expression, you are likely going to completely upset the cells that you are looking at, and you can really start to doubt how relevant things are that you are finding. And so that gets us to the second age, third actually, the age of gene targeting. And this is going back to this beautiful blastocyst. And as I said, it's a hollow ball that you find in mice in about 3.5 days of development. And these cells, it's again a very amorphous blob of cells. It's what's called the inner cell mass. And in the 1980s, it was found how you can take these cells out of the embryo and put them in culture in a dish. And when they're cultured in a dish, we call them embryonic stem cells. And these are the functional equivalent of these cells in the inner cell mass. I'm not saying they are identical, but functionally they are definitely equivalent. And the good thing with working with these embryonic stem cells, they are, as you will remember, my first picture of the life cycle, they are still before this point where we get the division of cells going into the actual animal and cells getting into the germline. But since we can culture them, even if all the manipulations that we are doing are relatively rare, and although mouse ES cells are extremely efficient in the type of things I will be talking about next, we're still talking about an efficiency, if we're lucky, of 1 in a million cells. But by doing this in vitro, we can actually invent tricks, again, steal tricks, to go from that very rare cells somewhere in a dish with millions of other cells to, in a couple of weeks, end up with only the cells we want. And this trick is called positive selection. And we often do this using genes from bacteria that give resistance to particular antibiotics. So what we're actually doing is, I should go one step back. Whereas the micro injection, what we discussed before, was a completely random process. With embryonic stem cells, we can actually make the precise mutations, variations, changes that we want to. And we can do that by making what's called a targeting vector, which has 2 particular things. First of all, it will have this antibiotic resistance gene. And second, it will have these what we call homologous arms. And this piece of DNA, we're not anymore talking about this bit, will go into the cells by electroporation or something like that. And through a process called homologous recombination, which is actually a normal cellular process that every single cell uses to repair the double-stranded DNA damage. It's not something we put in, it's nature is doing everything for us. But these homologous arms will find the same sequence in the target genome, and this piece of DNA will replace the endogenous part. So here we are making changes to the actual endogenous gene in this particular cell. And this construct has been made in such a way that this antibiotic resistance cassette not only gives this positive selection, but at the same time, it will also delete part of the gene and make the mutation that we want to make. So we're doing two things in one go. We can go from a single cell to a whole dish of cells, and we're actually mutating a gene as it is in that particular genome. And again, once we have these ES cells, we can start sticking them into embryos again. There they come. In the end, we're not injecting DNA, but we're injecting embryonic stem cells, and the embryo is a bit later. But the whole concept is, again, the same. And then once we've done that, again, we put them back into pseudo-pregnant females. And if these embryos that were injected with these extra modified embryonic stem cells are giving rise to pups that are being born, we get what are so-called chimeras. And this means that these are a mixture of cells that come from the cells that already were in the inner cell mass of the blastocyst that was injected, plus there are cells, materials, tissues, what have you not, that came from the cells that were injected. Now, we often use a trick by using embryonic stem cells that are derived from a mouse strain that has a coat color that we call chinchilla, which is light brownish, light grayish, whereas we inject them into a blastocyst from a mouse strain that's black. So these chimeras will literally be black with white spots, and the more white you have, the more the ES cells that you injected have given rise to these pups. And then once we take those chimeras and we start breeding with those, in the next generation, again, we can start to get animals that are completely derived from the cells that were injected. Now, the big advantages of this is literally everything is possible. And I'd like to make this perfectly clear. The fact that we can now make any possible modification that we can think of is not completely because of the genome editors. There isn't a single thing that so far I have seen that can be done, I should say in mouse, that can be done with the editors that wasn't possible before. It's only a lot more efficient. The positive selection can usually compensate for the high efficiency, and from an experimental biological point of view, an essential difference with the previous technique is that we can use to inactivate or modify endogenous genes. We're not just adding things, we're doing things with the genes as they are normally functional in the animals. We can also, by using a slightly different type of targeting vector, actually put extra genes into this animal, and then they will end up exactly where we want them. So even there, we will have much more control than we have by just injecting the DNA. And if we have more control, it is more predictable, and it will save a lot of animals. The disadvantages are it's only possible in species where we can use ES technology. This sounds like a minor thing, but in practice that basically limits it to mice. Red ES cells have been available since 2008, and indeed you can make mutant rats that way. It is being done, but it is still mainly mice that are being made this way. Second, you need this positive selection cassette to get to the efficiency you need. And this can potentially have an effect on the locus as you are making it. You can do all possible tricks to remove that again, but this takes extra time, both in making the construct and everything else. We need these large homologous arms, and large, it's about between 3,000 and 5,000 base pairs on each end, which can make it more complicated to make the construct, but it also complicates things considerably with actually finding whether a cell or a particular clone of cells that you get is exactly what you want it to be or not. The shorter these arms, the easier that is. And finally, you have to do these extra breeding steps, which will add an extra 6 to 8 months before you can actually start to generate your experimental animals. So this, of course, a time issue. It's also a number of animals issue. Yeah. So next way of making a mutant, and we're actually going back to the age of breeding, but let's call this the age of breeding with a little bit of help. And these are ENU screens. And this is a lot of breeding, and I'm not going to talk you through the details. This technique has been used in a number of species, mainly mice and zebrafish. And it works by taking a male and give this mutagen. Usually this is a mutagen called ENU. And I'm not going to tell you what it exactly stands for because I forgot. But what it does, if you use it at the right dose, and this needs a lot of testing and trying, but if you use it at the right dose, you will on average get 1 random mutation per gene. So you have a male or a few males that have all these random mutations in their germ cells. With those males, you start breeding, so you get offspring. And at this point, all these mutations end up in different offspring. And there you can, if you want, immediately start looking at phenotypes. This is sort of what we call reversed genetics. We start looking for phenotypes that we find interesting. And if you're working on deafness, you start looking for mice in the second generation that are deaf. If you're interested in blindness, you start looking for mice that are blind. And you can do all these phenotypic screens, and these can now be done on really massive, almost industrial scale, where you have all these phenotyping pipelines to simply find phenotypes. You can also, in this first generation, you can find mutations that, as we call it, are dominant. You only need 1 mutant copy of that particular mutation to see a phenotype. But you can also do quite complex breeding schemes to end up with animals that have both copies of this particular gene mutated. So you can also find recessive mutations. And so this has clear advantages. It's a very efficient way to make many, many different phenotypes. As I said, you can do dominant as well as recessive screens, and then it can and has been done in multiple species. Especially now with the next generation sequencing that is possible, it becomes manageable to actually find a mutation. 15 years ago, the difficult part was not making the mutant animals and finding the mutant animals. It was finding the mutation that actually caused it, because we didn't have easy access to the whole genome sequence. Now you can simply sequence for $1,000 a genome of the animals with the phenotype, and you're bound to find it in a couple of weeks, if not faster. And also important, there's the different ways that you can simply make all these models, but freeze them down. And even if you have backup material that you can use for sequencing, you no longer need to keep all those lines walking or swimming around until you find the mutation. The disadvantage is that it's sometimes difficult to really completely 100% prove whether a particular variation you found is actually the causative mutation. That's partially because there are often missense mutation that can be difficult to predict if it's actually a mutation or a variation. You need a lot of information about the host genome, which used to be a big limitation. These days, it's not that big of a deal anymore. But also, it takes a lot of animals, and especially late phenotypes can be impractical. For instance, if you're interested in cancer, which also in mice can take months or 1.5 years or longer before it's found, you need to keep all those animals on the shelf, and that's a lot of animals, a lot of time, and a lot of money. And in the end, although you can get many different phenotypes, you cannot say beforehand, I want this particular phenotype, I'm going to do an EN U screen and I will get it. Because in the end, it's still a chance process. And that gets us to the last couple of years, which is the beginning of the age of editing. And this is partially going back to what we already discussed before. But the idea behind this is that if in every single cell there is a double-stranded break in the DNA, the cell will have two major, there's actually a few more, but two major pathways within the cell that the cell has to deal with this broken DNA. Of course, you will understand a break in the DNA for a cell is not a good thing, so the cell wants to repair it. The simple way of repairing it is called non-homologous end adjoining, or NHEJ. And with this, some proteins in the cell, again, we are stealing this whole thing from what the cell already has. Some proteins there will recognize this broken DNA. They will bind to the ends. They will chew a bit off to make nice clean cuts and stick them back together. Very simple for the cell. The problem is, as I said, the enzymes chew a bit back, so you lose genetic information. And because of this will likely cause a mutation, because we're taking away genomic material. These are usually deletions, sometimes insertions, but in 2 out of 3 cases, this will put the protein in a different reading frame, and we will pretty soon run into a stop codon, and therefore we are making a mutation in precisely the gene that we want. We're not making exact the mutation that we want, but we are making the mutation precisely in the gene that we want. If we do want to control exactly what we are getting, we use the same homologous recombination pathway that we also used in the embryonic stem cells. And again, this is based on giving the cell an extra piece of DNA, which we call in this case the repair template. And the HDR machinery from the cell will use this template to copy it into the side of the break. And we are precisely repairing exactly the way we want the break to be repaired. So if we put in this repair template a very precise amino acid, change exactly the one that we want to do, we will end up with exactly that amino acid change in the repaired locus. So this gives us the same sort of control of things that we already could do in mouse embryonic stem cells. So the age of editing has seen different players, and although it started with so-called mega nucleases, which were almost impossible to work with and to make, they're now usually forgotten. But the first system that came available that was really useful are the so-called zinc finger nucleases, and this goes back to, again, natural zinc finger proteins which the cells use often to control the expression of endogenous genes. I think the zinc finger proteins might very well be the largest protein family in the mammalian genome. And these proteins have a particular three-dimensional structure which is being kept together by a single zinc ion, which binds to two cysteines and two histidines. And that way, this whole piece of the protein is kept in a particular three-dimensional shape. And this shape can actually fit into the double helix of the DNA molecule. And that way, the protein can sort of feel on the inside which particular nucleotides are on a particular place. And in practice, each zinc finger recognizes 3 particular nucleotides in the DNA. So this is a very precise way that a protein can read almost what sequence there is in the DNA. At some point, people started to realize that this is actually pretty handy, because you can start making engineered zinc finger proteins that just use these zinc fingers with that are defined by you to bind the particular DNA sequence that you want to bind, and you can sort of use that by coupling another functional part of a protein to these zinc fingers to actually get all these activities that you might can think of, to get them to the exact sequence in the genome where you want it. Again, as I said, it works fantastic, but we didn't make it up ourselves. And the important variant for this meeting were the engineered zinc finger nucleases, which indeed consisted of a few of these zinc fingers against the sequence that we were interested in, and they were coupled to a so-called restriction enzyme. Restriction enzymes are used by bacteria to cut the incoming DNA at a particular sequence, and this particular restriction enzyme is called FOC1, and this has the handy characteristic that it works in dimers. So that means we make a zinc finger with a FOC1 domain on one side and one on the other side, and only when these two come together, the FOC1 gets active and we cut the DNA exactly where we want it. Brilliant. Just what we wanted. Zinc finger nucleases were the first truly programmable nuclease system that could be used for genome editing. And importantly, it allowed the targeting of endogenous genes in other cells than mouse embryonic stem cells. There were two ways of doing it. The first was a commercial option, and this was incredibly expensive. You are talking about $50,000 for a single set of zinc finger nuclease. You had a pretty good chance that it would work, but most labs, including mine, could not afford it. There was an open source option, I would like to call it, which was affordable, but even labs who are really good in these sort of things, it took them at least 6 months to get it going. So in that context, it was still expensive to introduce it to your lab. And also because, in theory, the zinc finger recognizes especially 3 specific nucleotides next to each other. Sounds all nice, but in practice it means that the nucleotides around it also have some effect. And because of that, the pairs, the targets that you can actually find were relatively limited to what we are used to now. So we come to the second part of the age of the editors, and sometimes I think if the zinc finger nucleases hadn't been made so expensive, then other people wouldn't have been so eager to look for alternatives, and we might now only be talking about zinc finger nucleases. But the second system that was developed, and I think we're now, from the top of my head, 4 or 5 years ago, are the so-called TEL effector nucleases. And these are, again, proteins that consist of a DNA, a sequence-specific DNA binding part, and an effector domain, which again can be anything that you want to get to a specific site in the genome. This DNA binding domain consists of a lot of repeats. And these are repeats of 34 amino acids, which are almost identical, except for the 2 amino acids at position 12 and 13. And you have different variants there, and exactly the 2 amino acids that are on that position will determine whether this individual repeat will bind an A in the DNA, or a T, or a C, or a G, and sometimes an A. The system came, again, we stole it from a plant pathogen called Xanthomonas, and they actually use these proteins to activate, so they infect a plant, and they try to activate the genes in the plant cells that are handy for them for whatever reason. So they inject it into the cells, and that cell, that infected cell, will start to express things that the pathogen can use. But the important thing, there's a very simple one-to-one relationship between the particular repeat at a particular position and the target nucleotide that's being recognized. So this is this gives much more control, much more predictability than the zinc fingers. Everybody who ever tried to make to clone a piece of DNA will know that if you have almost identical repeats of 34 amino acids, and you have between 20 and 30 of those, and you want to get them in a single piece of DNA, it's hell. I mean, this will cost 3 or 4 PhD students the will to live, or at least the will to stay in science. However, somebody came up with a very elegant cloning system for specifically these sort of things. It's called Golden Gate, and with those actually in about 2 weeks, everybody could make these constructs. Once you had the system going, I've had people in my lab, undergrads, who had never seen a pipette, let alone touch it, and they could even make them in 3 weeks. Compared to the zinc fingers, it was complete new ballpark. There's also more targets, and importantly, compared to the zinc finger, they started using what I would call the academic-friendly licensing system. It's different for companies, but for academics, it means you can just use it. You don't have to pay your whole budget to be able to use it. And indeed, all the reagents, they were started to be distributed via AdGene. AdGene is a not-for-profit plasmid depository, where everybody can simply order for $60 and a few dollars shipment all the constructs that you want. And the people who developed these systems immediately put everything there, so everybody had easy and affordable access to it. So everybody could start testing it, everybody could start thinking of new things to do with it. At that moment, it started to take off. And indeed, just as with the zinc fingers, it can be used in many species of cell types. But this time, people could actually afford to start using it in different systems and different cell types. And then we got to the third part of the age of editing, the CRISPR-Cas9. And again, a little bit of biological background. Of course, evolution didn't give us CRISPR-Cas9 to make mouse models. It's the adaptive immunity of bacteria, and the way it works is that let's say the immunity with memory, like our antibodies. And pieces of DNA that invade the bacteria somehow end up in the bacteria's genome, and these are being expressed again and then picked up by the Cas9 nuclease. I'll show some more details in a couple of slides. And this Cas9 is a nuclease, so based on these pieces of RNA that in the end, sequence-wise, are coming from this pathogen, there the incoming DNA or RNA is being cut up. This actually goes back quite a long time. This whole idea was for the first time described the existing of these CRISPRs in 1987. It was the year that I finished high school, and I checked it. The biggest hit in 1987 in the UK was Rick Ashley, Never Gonna Give You Up. It took until 2013, after which, of course, we all had seen Rick Ashley many times via the Rickrolling. By 2013, the whole mechanism behind it was elucidated, and people started to realize that they could use this to cut any piece of DNA. And this is indeed how it works now. We only need, in principle, two things: the Cas9 nuclease protein and a piece of DNA that you can turn this into very complex figures, but in the end what it does is the piece of RNA tells the Cas9 nuclease where to go and where to cut the DNA. And it's as simple as that. So if you would characterize CRISPR-Cas9 with 2 words, it's extremely and it's many. Because it's extremely easy to clone these constructs, and many at the same time. You can do it in an automated manner without any problem. It's extremely cheap because, again, you can get everything from AdGene for a few dollars. It really works. Often you see those papers on a new technical development, and you think, yeah, let's wait a couple of months before starting to use it and see if other people can also do it. This simply works the first time you do it. There are many target sequences, even many more than for the TAL effectors. And again, here people started to realize that they can use this to get all different types of activities to that particular site in the genome. And many, if not all, species can be modified this way. So I'm not going to talk you through all the details, but if you indeed compare zinc finger nucleases to TALENs and CRISPR-Cas9, it's pretty clear that for almost everything, CRISPR-Cas9 is the way forward. And then, of course, we can use this to make mutant animals. And this is the same figure I showed before. And we can use these NHEJ, non-homologous end-joining induced random mutations where we want them to simply do a quick knockout of a particular gene of interest. And we can use the homologous directed repair pathway to make the extremely precise mutations that we want. So we can use it in two different ways. First of all, you can simply use it in embryonic stem cells, the way we were always doing things in embryonic stem cells, but just make it much more efficient. We can make the arms, the homologous arms, a lot shorter, and because of that, it becomes lots and lots of easier and faster to screen all the different clones to find what we need. And with 800 nucleotide arms, we and other labs routinely find efficiencies of 40 to 50%, which is compared to about between 1 and 5% the old way. We can do targeting without positive selection because it's so unbelievably efficient. So again, it makes things faster and better to predict. We can make small mutations or variation, for instance, epitope tags, by using simple oligonucleotides that you can just order from a DNA synthesis company that you will have 2 or 3 days later if they are really slow. And with that, without having to make any sort of other targeting vector, you can do what you want. But also, and that's something that we are doing a lot at the moment in my lab, because it's so efficient and so fast, we can do a lot of step-by-step modification to get incredibly complex alleles for our work. Things that if you would have to do it the old way, you would simply not even start with it. But the most important thing is that several labs, including the lab of Rudolf Janisch, whose work I got this picture from, they show that you can take these nucleases, CRISPR-Cas9. This had already been shown for zinc fingers and tau effectors, I should say, and simply inject them in the fertilized oocyte, as we did with these old-fashioned DNA constructs, and it would work. So we would have the speed back that we had with the simple DNA constructs, but now we could do all the precision editing that was needed. And indeed, a quick figure from a table from the same figure, it is incredibly efficient. The biggest problem at the moment is the relatively low efficiency of getting bigger pieces of DNA exactly where you want it, but everything else goes incredibly efficient. So this direct oocyte microinjection, that basically gives us the control of the models that we want to make that until now was only possible in embryonic stem cells. Therefore, for most species, it was not possible. The only exception, mouse and being mouse and a little bit of rat. And for this reason, for several decades, if you needed this sort of control over your mutant animal for purely technical reasons, well, the mouse was the only thing you could do. That's no longer the case. Everything where you can inject oocytes, and that's almost everything, you can do with that. And this is, in my opinion, why this is really a massive, has a massive impact on the way we use animals in science. Now, a little bit on the off-targets. I think everybody will have heard about that. With off-targets, I mean you are mutating the site that you want, but in addition, you're also getting mutations at other sites. And in principle, this is a risk for all the editors that we've spoken about. But CRISPR-Cas9, which is the most efficient one, was believed to be more risky because of 2 reasons. First of all, the actual target sequence is that is being recognized is a lot shorter than for the other system. So purely by a chance, it is more likely you find somewhere in the genome a site that looks like it, but also the other 2 systems use a double system to get the FOC1 enzyme together. Cas9 only works as a monomer. So because of that, it was believed to be a lot worse for off-targets. And indeed, looking at pools of cells and other ways, fancy ways of measuring double-stranded DNA breaks, that all seem to support this notion. And for some time, people really started to panic. However, of course, there was solution. First of all, by now, all over the web, you will find these in silico tools that will find the best possible RNA sequence to use in your system. And this already has a big impact on minimizing the off-target. Simply use a good design tool. The first variant that was made is the so-called double nicking, which is a mutant version of the Cas9 nuclease that doesn't make a double-stranded DNA breaks, but it only breaks or nicks one strand of the DNA. And by doing that at two close sites, you actually also, for the Cas9 system, come to a situation that you need to get this double hit close to each other for the system to work. It was also shown that the target RNAs, the guide RNAs, if you make them a little bit shorter, you reduce the cutting at off targets without affecting the intended target, which gives an advantage. And just last week, there was the publication of this enhanced specificity Cas9, which was really rationally designed to improve the specificity. It's, in that respect, it's a really beautiful piece of work. But also, and maybe this is only recognized by people who have been making mice for a long time. You don't actually need genome editors to find unexpected things. You just need to look for them. Because I don't know if I'm allowed to say this in a webcast, but in the end, shit happens. Two examples from my own career of labs where I worked. And either I'm incredibly unlucky with these sort of things, or it is a lot more common than we all think or want to admit. One example was a transgene, a random integration, so the very first system that gave a mouse with extra digits, polydactyly. This mouse mutant was called Sasquatch. And once these people indeed identified what the problem was, they found that this transgene had integrated in a regulatory sequence that controlled the expression of a gene called sonic hedgehog, which is a very important developmental gene, and it does this from a big distance. But once they found that, people could start looking in human families with polydactyly, and they found mutations in exactly the same element. So here, a random thing in these mice tells you what to look for in humans, and you will find mutations in these families. Fantastic. But also elsewhere in evolution, do people know the Hemingway cats? Ernest Hemingway had got at some point, I think, a few cats, and one of them had extra digits, and they were just walking outside and breeding and breeding and breeding. And now if you go to his old house, that there are dozens and dozens of cats with extra digits. These are called the Hemingway cats. Here's one of them is extra standing next to his original typewriter, and this is an incredibly cute one. So again, here it happened because of the modifications. Here it happened completely random. Nature does this itself. But also my own PhD, actually, I was working on making a mouse model for breast cancer. In our embryonic stem cells, we found, in hindsight, that we picked up a single purely random chance nucleotide change that gave the mice intestinal cancer. Didn't have anything to do with the BRCA1 project. It just happens. And it gives you interesting phenotypes. This was all without editors. Now we would call them off-targets. It's now also clear that by actually sequencing the whole genome of edited cells and edited animals, that it looks like you have a bigger risk of these off-target mutations, but you don't actually find them. And because of this, in animal work, the off-target mutation might not be as big of a worry as we all thought 1.5 years ago. Of course, in human editing, this is a completely different story. But in practice, just using good design for your guide RNAs will get you pretty far. So finally, very briefly, what does this tell us for what impact will this have on the ethics of using animals? And this, of course, I'm doing around the 3 R's, the replacement, the reduction, and the refinement. So can genome editing replace some animal research? And I think that's definitely the case, because we can start to do a lot of things now in cell lines that until now could only be done in animals, because we could only do it in mouse embryonic stem cells, and we had to turn them into animals before we could actually do the things. Also, because until now we could overexpress things in cell lines, we could use RNAi, but that's only a partial loss. There's now a whole new world of experiments possible in cell lines that were not possible until now. And I think especially in combination with the recent developments in taking human embryonic stem cells or iPS cells and differentiate them in vitro to the different cell type, different tissue, I think this is the killer application that could potentially reduce a lot of animals. So can editors refine animal research? It looks like a clear and obvious yes, but I think there are some nuances there as well, because as I said, until now many types of research had to be done in mice, simply because it was the only place where the only species where we had the technology. So now we can start using other species. Lower species. I think we're not supposed to say that anymore. But for instance, zebrafish is going to be extremely interesting for this. Then again, for other projects, you might actually want to go to a higher species that until now was not possible. But a pig might simply, for the thing you want to model, might simply be a better model. And I'm sure we will be hearing a lot about that, both of these things later today. But in the end, whether you or not you can do something is only one part of why you are using a particular system. Using a pig is a lot more expensive than using a mouse. If I don't have access to a facility to work with non-human primates, I'm not going to do my project in non-human primates, how important it could be. There's a lot more that comes into place. And finally, can we reduce animal research? And that, I think, becomes really tricky to predict, because if we actually look at the number of animals of the particular species that are being used, and this is all data from the UK Home Office from 2013. 82% of the research was done with rodents, mainly mice, and then we have 12% fish. The rest is small. It's very small compared to these species. And within these species, over time, you see that the increase in animal use, it's actually just down to mice at the moment. Rat and zebrafish, rats going a bit down, zebrafish going a bit up. But what we are talking about in the end is mice. So if we look at what sort of mice are being used, normal animals has been going down, but these are not that directly affected by genome editing. They might go down a bit further because there's now possibilities to make better models. So if it will go down because the use of normal mice will go down because of the editors, that will be at the price of an increase of the modified animals. At that point, there was still a distinction between harmful mutants like ENU mutants or random mutants and modified. The EU at least has now combined these. But this is actually where the numbers are going up. The genetically modified mice. And what are we doing that is giving this increase? We are breeding. Just everything else does not change at all. With better models, it might go up a bit, but the big increase in animals these days, it's down to breeding. Often, if you are lucky, 1 out of 4 mice in your litters is the one you actually need. 3, you are getting them because of Mendel. You don't need them. You need a few controls, but we're getting way too many controls. Can this be changed by editors? And that's I think my last slide. Maybe, but it will take a big change in the way of work. And there was this paper from the, again, from the lab of Janisch, that showed that they could, by simply injecting different guide RNAs with Cas9, they could directly in oocytes make completely knock out 2 genes, 4 alleles in a single animal without any breeding. In ES cells, they knocked out in one go, in a single experiment, 5 genes. And they cheated a bit because 3 of them were on the Y chromosome, so there they only had to knock out 1 copy, but still they hit 8 alleles in one go. Now, it is technically possible, but there's not many places who actually do it to not make chimeras, but use what we call fully ES-derived mice in one go. And this combination, I think, might get rid of a lot of this surplus breeding problems. But it would require that everybody's starting to work on a completely different way. I actually think it might also be cheaper, which would be another big advantage or a big stimulus of the changing the habits. But the most important message I think from this, that if we start using more species, we really need to make sure that if we can prevent us from getting into this same surplus breeding problem, we should definitely keep an eye out on that, because I think there is where the big gains in lower numbers are possible. So finally, a little bit of an overview. Yes, I think genome editing will have a big effect on the way we use animals in research. But beyond that, it's difficult to predict what will happen, I think. I think initially things will go up. Everybody will need to learn how to use these techniques. And maybe there will be a little bit kid in a sweet shop problem. People, things they couldn't do before, they might want to do now simply because they can. And that's, of course, something that needs to be checked by the local ethical systems. After that, numbers can go up, numbers can go down, the species might shift, they might not. I really don't have a clue what in the end will happen. And also, if making models becomes easier and cheaper, if I can make a new model for 10% of the cost and 10% of the time, my funders do not expect me to start rolling my thumb for 90% of my time. We will do 10 times as many projects. In the end, I predict we will use the same number of animals, but we will get much more scientific output from it. And I think that's something that we might have to take into account when we are talking about these numbers. It is not just numbers. It is numbers in the context of scientific output. And finally, what I called the reviewer 3 problem. Why don't these authors quickly make a knockout for this particular gene, whatever one, and see what it does? I think it will become too easy to make animals, not even if you want to do them themselves, but you're basically being told, and again, you need to really keep that ethical focus and ethical review system on top of that. So finally, although I didn't talk about my own research, I do want to point out these funders that have been putting money into my lab over the years, particularly at the moment, University of Edinburgh, the MRC and the BBSRC, who are all paying a bit of my own salary. And with that, I'd be happy to take questions. It depends on the scientific question. Whether the use of editors has a particular advantage or not when you're using recombinant inbred strains. I think when you have a pure, clear scientific question, I want to know how this works or that works. In the end, as I said, I don't think the off-targets is not what keeps me awake at night. I've seen the data of people that backcrossed the same backcross, two different mouse lines for 10 generations on the same, and then they started sequencing the whole genome. So nothing happened in between. The changes were tremendous, more than 30 inactivating mutations. So I think, yes, that is for a clear answer. I think it will be a big advance there because you will get them in the same background. For modeling disease, for really preclinical models, I'm actually starting to think that recombinant inbred strains could be a disadvantage in the sense that, yes, we can use fewer animals, we can get easier a clear answer, but we only get a clear answer for that particular genetic background. And if you are using that for preclinical models, you are not modeling what's actually happening in your patient population. And for that, you might actually want to use these controlled outbred populations. There, it will be a lot more difficult to edit. Well, it will not be more difficult to edit them, but because you then have to start breeding, you're actually inbreeding again. So there, it will be, in practice, a lot more complicated.
Thanks for your talk, Peter. You stated it's something I wanted to follow up on about ENU mutagenesis, and you said it's really hard to tell the difference between a mutation and a variation.
Yeah.
Could you define what the difference is?
We can do that over a couple of beers. No, I would almost say, okay, the easiest way of saying it, I would call it a mutation if it changes the phenotype. And that's an incredible oversimplification. And then you would only call it a mutation if it changes the phenotype that you're actually analyzing. And so if you don't see things, and I would, based on that, call it a variation, we might simply be looking at the wrong phenotypes. It is becoming a very gradual difference. I think that might actually be what the people working with human genetics and large genome sequencing projects, what keeps them awake at night. Yes. Can you wait for the...
No, it's for the web.
Does off
It's on.
It's on.
It's on.
does off-target risk vary with the animal, like between mouse and monkey?
Not aware that there's any data on it published. I would be surprised. But then in the end, I think you need to answer that question by really just sequencing the whole animal and not look at all these surrogate types of analysis, because in the end, they didn't give the real answer for mice. I don't see how they would do that, thank you, for other species. But I would be surprised. Nothing more? Okay.
Thank you very much, Peter. It was a very insightful talk. And a great background for the rest of this morning session. But before that, we'll be taking a break, and if everybody could be back by, I guess, 10:15, that would be great. Thanks a lot.